So we know that they belong to a class of enzymes that is called nucleases, and there are two different kinds. That is exonuclease and endonuclease. So from the name itself, we can say exo means from the exterior of the ends, endo means within, right? So they remove the nucleotides from the ends of DNA, so they are called exonuclease. And endonucleases, they cut at specific positions within the DNA, that is in the middle of the DNA sequence. They bind to specific recognition sequence of the DNA and cut the two strands at specific points, right? So for example, this is the action of the restriction enzyme EcoR1. So this is the recognition sequence, right? GAA-TTC, and this is the cut site between G and A. So the enzyme cuts both DNA strands at the same site, that is between G and A. The EcoR1 cuts the DNA between bases G and A only when the sequences GAA-TTC is present in the DNA. So wherever the sequence is present, that is GAA-TTC is present, then the EcoR1 recognizes that particular sequence and it cuts the DNA between G and A. Okay, so this is as a result of the action by EcoR1. So now what happens? So here these nucleotides are removed, so it forms a sticky end. And here also in the case of foreign DNA, it has the same, that is the recognition sequence. So here also it is cleaved by the same enzyme EcoR1 and it produces sticky ends. Now these two sticky ends, that is one from the vector DNA, the other from the foreign DNA, and they are joined together, right? So they are joined together by means of the enzyme ligase to form a recombinant DNA. So this is a recombinant DNA, right? So the main difference between exonuclease and the endonucleases, exonucleases cut from the ends of the DNA, but endonucleases cut within the DNA.